Hi, this is Miss Litton, and this is lunchtime, and I have some wonderful AP biologists here. Say hi. Oh, that was weak. Okay, and we are on finishing up our first big idea on evolution, and this is 1D. The origin of living systems is explained by natural processes. So first thing I want to show you, I don't know that they'll ask you this, but there are two strategies for how you would approach the study of the origin of life. So there's the top down and bottoms up, which are exactly what they sound like. So you do a deconstructionist model, breaking things down, and then end up going to the smallest bit, which is mapping it using finding Luca. Okay, another option, bottoms up, is pretty much how I taught it to you, where we looked at the smallest molecules. We said, okay, where could these organic molecules have come from if we had an inorganic environment, right? And then we looked at that, and then we looked at how do we make polymers out of monomers. Remember how we worked our way up? And then the first cells and so forth. So there are two strategies. Underneath bottoms up, you have two ideas. Did metabolism come first, like the process of cellular respiration come first? Remember we talked about, would an organism be an autotroph or a heterotroph, do you think? Which way would come first, autotroph or a heterotroph? Are you mumbling while you're eating heterotroph? Yeah, because it would be easier to be a heterotroph because you consume nutrients. If you're an autotroph, you have to produce the nutrients and then break them down. So that would be a more difficult thing to evolve. Okay, so then there's the idea, did the metabolism come first or being a self-replicator come first? And remember we said, was that first genetic code, do we think it was DNA or RNA? And we came up with some pretty good arguments about why it would be, what one? RNA first. Because RNA can act to store information, and it can also act as a what? Remember? As an enzyme. Perfect. Okay? So anyway, those are the two approaches. We did the first one. So we talked about how old the Earth is. Oh, there it goes. Okay, maybe around 4.6 billion years old. And we talked about that early atmosphere. It wasn't an oxidizing environment, but a reducing environment, which meant it had no free oxygen. Did it have any oxygen? Yes. Wrapped up like with hydrogen, H2O or carbon monoxide, right? So it's not that the atom is not there. It's just not free oxygen. Now, there was a benefit to that. Do you remember what was the benefit of a reducing environment? Yeah, nothing to break things down. Nothing would get oxidized. So it was conducive to polymerization, for instance. You could build because things did not tend to degrade and oxidize in the presence of oxygen. Okay, so, and those first gases could have been things that came out of like a volcano. It would be very similar. So we talked about that primordial soup, and we said those early gases, and you had rain that came down, and you would have this mixture of molecules, and we wanted to form organic molecules and monomers and eventually polymers, and we talked about how could that have come about. So, oh, I went a little bit quick. Sorry, but here are the gases that you would first have, no free oxygen. Okay, and then we put it as like a stair step. So first thing we have to do is take those inorganic molecules that could have come out of a volcano and try to get some organic molecules. That's step one. Do you remember how we got from the first step to the second step? Could have been lightning or molten magma, some sort of energy, sun's radiation, and that energy would be enough in high concentrations of inorganic molecules to make these organic molecules. And in fact, a scientist proposed that, starts with an O, Oparin, and then a different man did an experiment. You have to be 21 to drink it. Miller did the experiment. He's drinking some primordial soup. You're like, oh, that's what I drink every day for lunch, Miss Litton. Miller did that experiment. Remember he cycled everything through? Do you remember that part? I'll show you some pictures. So, hi, is there something here? There we go. there's our inorganic molecules, and he was able to get simple organic molecules. And then we have to go take a step up from these organic molecules that are simple, like simple sugars, and we need polymerization to take place where we form complex macromolecules. What are our 4 organic molecules we want to make? I can't hear you. Carbohydrates?
Nucleic acids.
Oh yeah, nucleic acids, and what did you say? Proteins and lipids. What are the 2 types of lipids? Fats and steroids, right. So once you have your amino acids, how do you get the polymer? How do you get those proteins? And we talked about how clay could act not in to facilitate the reactions. We talked about the high concentration. We talked about proteins. When hot proteins cool in an aqueous solution, they can even form the beginnings of what would almost be like a cell membrane. So the energy in the environment would be enough. Then once you have those macromolecules, you have to build cells. That first cell, that cell membrane, it's going to keep whatever's going on inside is different than what's going on outside. Okay? And was the first cell probably prokaryotic or eukaryotic? Yeah. And once you had a self-replicating molecule that was able to store information in some sort of nucleic acid, then you had what? Life. And so you want to be familiar with those arguments as you work your way up. If we look at what the next slides are, is basically what we just went over using the ladder, how we went and how we could, or up those stairs up, how we could build monomers. Again, we talked about energy from the atmosphere, hydrothermal vents, and maybe comets brought organic molecules here. That would be a possibility as well. Once we had our monomers, we talked about this. We looked at those monomers being formed in Miller's experiment in just a matter of a couple of weeks. He was able to form those simple organic molecules. Then for a polymer, again, we talked about protein first and RNA first. Do you remember that argument and why? Because if you're going to do polymerization, we're used to doing that with some sort of catalyst or enzyme, right? So you don't have one. Proteins are the enzymes, right? But how do you make the proteins if you don't have the genetic code that codes for the proteins, right? So that was the argument, and that's how we went with the whole RNA first, because it could do both. Okay? Once we have our proteins, nucleic acids, we have our polymers. We talked about proteins first, Sidney Fox. We talked about using clay, and that maybe they were generated simultaneously. Clay can act as a catalyst, or maybe RNA first. We just went through those arguments. Okay, now your first cell, autotroph or heterotroph, and we already talked about this. You would probably have a heterotroph first. And what type of respiration would come first? Anaerobic, good. And so what step would that be? What would be the first step? All cells, what was that? Glycolysis followed by fermentation. Okay. All right, and so that's this part right here. Okay, there are no organelles at this point because you're just a prokaryotic cell. So these are happening across membranes. All right, turn to the person next to you on just what I've discussed. What are some things you might need to review or look at? Go ahead. Don't say everything. Be specific. You know some of it. All right, can I keep going? You all right? Stressed out? Are you good? Good? Okay. So this is our information pathway. We feel pretty. This is the way we look at it now. We use DNA to code for RNA. We use RNA to code for protein. But it may have been that RNA was the originator, and DNA was just a good way to store it. Why? Because it's not single-stranded, it's double-stranded. It protects it, and we can store it in the nucleus in that way. but it's really RNA that gives us then our proteins. Remember we talked about it can code for other things that are really critical. It could code for transcription factors or chaperone proteins that change the shape and the purpose and the functionality of other proteins. We talked about the importance of spacers and exons and introns, right? And genetic recombination, all of that. All right. And so when we talk about the pathway of life, early earth, inorganic, small organic, polymers, early cells, self-replicating cells. That would be the pathway you'd want to be pretty familiar with. All right, now let's take a look at this. Okay, this is called horizontal gene transfer. Think about it horizontal, whereas parent to child is vertical. Discuss with your bio buddy, how could you explain that? It's either horizontal or lateral is what they talk to. Gene transfer versus vertical. Go ahead, have that discussion. Okay, then are you familiar with these three types of horizontal gene transfer? So transduction is when you use a virus or a phage, right, to move DNA from one to another. I believe I asked you a question. Did I ask you a question about this in your ed puzzle? Or is this something else? Who's done the ed puzzle, the review? Didn't I ask you? It was a model, and I asked you to explain it? Yeah. Okay, good. Transformation, this is what you're doing in your genetic engineering lab right now, is a form of artificial transformation, right? Because you're going to change an organism's traits by giving it the DNA that you genetically engineer. And then conjugation, what would you have to use between these two to make that happen? A sex post, right? So this is all horizontal gene transfer as opposed to the vertical. So I want you to take a look. This is, I know, a wall of words. There are 7 items on here. I'd rather, instead of me explaining it to you, explain to somebody next to you. So break yourself up. Whoever's in the slate seat, you take care of all the odd ones, and whoever's in the blue seat, you take care of the evens. Walk your way through. Make sure you understand it without me.
But like most of the time, horizontal genes most commonly it's found in prokaryotes, but some unicellular also do now artificial
Do you see an essay question right there from them? You know, compare and contrast horizontal gene transfer and vertical gene transfer and its role in evolution. Okay, that to me would be a great essay question. When you look at vertical gene transfer, think about that. It's not only what genes that got passed on, but what genes didn't get passed on. And when we talk about natural selection, right, those organisms that are best adapted to the environment are the ones that are surviving. Those are the ones that are the biggest, the strongest, the fastest. They're the ones who are passing their genes on. That's one way. And that can be directional selection towards the best adapted. But you can also have vertical transfer through genetic drift, right? Or the 2 types of genetic drift. Bottleneck and founders, right? Nature didn't select there. That was random, right? That's why it's called genetic drift. And that is, most of your evolution is on the vertical. But you can also change on the horizontal as well. And these are 3 good ways to do evolution on the horizontal. You good? Okay. And then, I have some questions for you. Let's pick an evolution word. I'm going to pause this while you guys log in. I don't remember how many questions I have here. Okay, so let's go through your questions. Yay, good job on the first one. So let me go over this in case people who are watching the recording can't hear it. The atmosphere in which life arose lacked oxygen. Which of the following is a reason why life does not arise today? There is an oxidizing environment, so it breaks down. The person who performed the experiment that proved that amino acids could be produced in a laboratory from a reducing environment was Miller. Good job. And protocells probably obtained energy as heterotrophs. Perfect. Oh, and the last question. Who was it? Weston, was it your bio buddy?
Are you throwing me under the You're so innocent.
Prokaryotic cells are represented by fossils that are dated back as far as, and 9 of you got 3.5. Some of you said, what did you say? 2.5, no. Okay, so round 2.3 was the what? Oxygen revolution. Yeah, that's when you're able to generate oxygen, which then made it so you could do aerobic respiration. All right, so let's keep going. Fossils. When you looked at dating of fossils, and fossils usually have to be about how old to be considered a fossil? 10,000 years old, right? Remember there was relative and absolute dating. Relative dating is when you look at the layers, right? And you assume the oldest layers are on the bottom. Obviously, you have to look to see if there's a subduction zone. Okay, but oldest layers are on the bottom relative to newer layers on top. Also, you might be able to age an actual layer. You're going to need to not talk, okay, while you're visiting. Look, he's not even hearing me. No talking. And so on, if you could date one layer, then you know every layer below that is older and every layer above that is younger. And then on absolute dating, you're doing how fast does a particular isotope or unstable molecule break down, and you know the rate of decay. And if you have a sample, you would expect half of it to decay. Like if I did it like this, if this was my sample, half of it would decay as compared to the total volume here, right? That would be a half-life. And then when another half of that decays relative to the rest of it, that would be your second half-life, right? And now the percentage is even smaller because it's had another half-life. So if I find something that is this percentage on the whole, I can go, oh, that's 1, right? 2, 3, 4 half-lives, right? And if that half-life is 5,700 years, then I just take 4 times 5,700, and I would know how old this is based on the percentage of that particular isotope found in that sample. Okay, turn towards your bio buddy and see if they are feeling comfortable about half-lives and rate of decay. Go ahead. Got a math test on it? Excellent. Okay.
Oh, Johnny, I thought you were telling me it was up already.
Okay, and then this is for perspective. Do you remember how we talked about the pre-Cambrian time was from the start of the planet, right? Remember that? From 4.6 billion years ago to about how many? Okay, so let's go over the different periods. Okay, so right now, the center of your world, we call it the Cenozoic, right? And it's this second back about how many? 65 million years ago, because you are driving and you drive about 65 on the freeway. Okay, then before this, the Cenozoic was the Mesozoic in the middle. Okay, and it ended 65 million years ago, but it started about what? 225, somewhere in there, okay, millions of years ago. Then before that was the Paleozoic, and that maybe started 570 million years ago. Before that is all Precambrian. And that's because the very first period of the Old and Paleozoic, that era, is Cambrian, so they say everything before that is Precambrian. Okay? So when you look at the bulk of time our Earth has had, look at this, on time-wise, because we know we're around 4.6 billion years ago, and the Paleozoic era was, remember how we remember it, like around dinner time, right? Look at how many million, like Precambrian, 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 oh, Paleozoic, right here at the end, for perspective. Okay? Paleozoic had the biggest jump in life forms. By the end of the Precambrian, you had simple invertebrate organisms. But during Paleozoic, you went from algae all the way to the beginnings of gymnosperms. That is a huge hurdle, right? You went from invertebrates to almost the beginning of reptiles in the Paleozoic era. So though that era is old and pale, huge jumps in life forms. And then when you look at the Mesozoic, as far as animals go, you know about it because of the movie Jurassic Park. What was around a lot during the Mesozoic era?
The dinosaurs.
Yeah, all the dinosaurs. And then this is when gymnosperms were all over the place. And you at the end of it, you had the beginnings of, yeah, what do you call those? Angiosperms. Okay? And so you have, and then in animal, as far as animals go, we went from reptiles into the early, by the end of the Mesozoic era, we had the beginnings of mammals, and then mammals, you know, were the dominant organisms in the Cenozoic era. And then right here, at the very last 30 seconds, is when probably man came about. All right, and then we looked at stromatolites. These are big bacterial rocks, okay? Basically is what you want to think of them as. And they were during the Precambrian times. They exist even now. So sand gets caught in the mucus of this bacteria. You have calcium carbonate from the seawater. And basically the living organisms keep moving to the edges, to the edges, to the edges. So just like you could cut down a tree and look at the rings of life, you could cut down one of these and see bacteria going back through the millions of years. And they've built these giant rocks. Okay? We've talked about the endosymbiotic hypothesis about 1.7, 1.2 billion years ago. And that was the formation of eukaryotes. So first you have heterotrophs, right? Then as a result of the oxygen revolution about 2.3 billion years ago, you have what? Autotrophs. And then this lends itself, the endosymbiotic hypothesis, to eukaryotic cells. And then you could have multicellular organisms, especially as the oxygen became more prevalent, because you could be large, multicellular, and have enough oxygen to survive for your needs, but also enough oxygen gave a what kind of layer? Ozone layer to protect them out on land, right? Okay. I know you're thinking all of those things. What is some evidence for the endosymbiotic hypothesis? Chloroplasts and mitochondria have their
Separate DNA.
Their own DNA, double membranes, ribosomes like a prokaryotic cell, self-replicating. And maybe they were once free-living prokaryotes which went to live inside of another prokaryote. Okay, and this, I don't even know, something's over there. Here's Precambrian, here's Paleozoic. These are the different periods. We've already talked about it. You went from marine algae to the beginnings of gymnosperms. We went from invertebrates to the beginning of reptiles. 3 massive extinctions. Now, extinctions could be bad because they wipe out a bunch of life, but it gives room for new things to evolve in their place as well. What kind of evolution is it when it goes out like a wheel? Adaptive radiation, remember that? Okay, good. All right, so here is the Paleozoic era, some of the changes that occurred there. Also during this time, beginnings of coal formation, compression. I already talked to you about the molecular clock. You don't measure time in seconds, but mutations. And there's some mutational rates, and then you could calculate how long ago two species diverged. And then this is showing you the Mesozoic, another couple of mass extinctions. And then here in the Cenozoic, just a significant mammalian extinction. And then maybe we're causing another one too. Yay. Beginning of primates. Here, oh, we're almost done. This is just showing you some of those big extinctions. Continental drift. More mass extinctions maybe due to rising tides, glaciers melting, changing in CO2 levels. More mass extinctions. More questions, but I know you need to go. 1 down, 3 to go. Good job, you guys. 1 big idea done. We just have to review 3 more. I'm super proud of you. Okay, that's it. Yay! You did that in 26 minutes.